Okay, so let's begin. Today I'll be talking about the investigation of metabolic phenomena using information theory. And currently I'm working with Pablo over here from the computer science department at Purdue, Mohan from statistics, myself, I am from the chemical engineering department. We're also collaborating with Max Roginski, who's a faculty member at UIUC. And so the general idea to begin with is that in my research group, we've been developing metabolic models, and this class of models is based off of cybernetic control. And so they've been very effective at describing dynamic metabolic phenomena based off of principles of mechanistic causality. And so what we would like to do is look at those models under the lens of information theory to gauge how accurate they are at describing real biological data. And so I prepared this presentation with the understanding that a lot of you might not have a strong background in biology, so I'll try to keep it simple so we can all be on the same page. And so to begin with, what is metabolism? And commonly we think of metabolism as a process by which we take in food and then convert it into energy, just like when we ate our breakfast today. But it's a little more extensive than that. So we do have something, it's fundamentally a chemical process. And so you take molecules that are relatively complex and then break them down into more simple ones. And in that process, you pull out energy. And in the other directions, you can take maybe the same molecule, which in the scheme of this pathway is less complex, and then use that energy to make more complicated molecules. And so this is, like I said, a chemical process, and it happens in different chemical reaction pathways. So this example is of glycolysis, where we have this same sugar being broken down in that process of pulling out energy. And so this happens on a pathway of multiple chemical reactions. And so these pathways interact with each other and form a network. And so these chemical reaction networks are rather complex. So what I want you to realize is that this window right here is a small picture of the overall picture of metabolism. And so these chemical networks operate in order to drive all processes of life. And it's a fundamental process that underpins life. So metabolic regulation is what we're interested in. Metabolic states are not static, and so if you have if you eat carrots for breakfast versus eggs, your metabolism is going to behave differently. And also, if you haven't had anything to eat for a while, your metabolism is going to be slower than if you've just eaten. And so what we're interested in is looking at this network, seeing how different control loops are monitoring the network, and how those create certain actions to change the state of the network based off of the biological needs. And so what I'd like to do is take a break and present you just a generic scenario. Let's say that you're flying out from Stanford and your plane crashes over the desert, and you haven't eaten for an entire week. And so a rescue crew finally finds you, and they present you with with several food options. Okay, they give you this, they present to you this nice salad, or maybe something heavier like a hamburger or a pasta if you're a vegetarian. And odds are that you're probably not interested in eating the salad. And so the point that I'm trying to make is that not all food sources are equal, and we can see this on a single cellular level. So looking at small cells like E. coli, they also regulate their metabolism sort of based off of this survival goal, right? Like the pasta is more nutritious than the salad. And so what we're interested in is how these systems have optimized their metabolic states through the course of natural selection. And so there's certain metabolic control strategies that have been filtered out that are more optimal for survival than others. And so in this context, certain sugars are more attractive to E. coli cells than others. And so all of these individual control actions converge on these greater control ideas, and that's what we're concerned with studying. And so, for instance, let's say we have the E. coli growing in a solution of both glucose and xylose, and so this axis represents concentration. This represents the concentration of the cells. And so there will be two phases of growth in this scenario. You have one phase where the cell is consuming the glucose first, and then another phase where the cell is consuming the xylose. And so this system is what cybernetic models of metabolism have been very effective in investigating. And so what our interest is after applying these metabolic models to a large variety of systems, we've met a lot of success. And that's based on this theory that cells are trying to maximize their carbon uptake rate. And so I'm not going to give an extensive picture of the models, but I just want you to get a general idea of how they work. And so they're based upon differential equations, modeling the various uptake rates and growth rates of the cell. So this would be uptake of sugar, the conversion of sugar into more cells, and then the creation of enzymes to digest those sugars. And so within these differential equations are certain control ideas that we model cybernetically. And so these cybernetic control laws compare various carbon uptake rates and then modulate the behavior of the differential equations towards certain metabolic goals. And so doing this, in that same scenario where we have glucose and xylose, we can see how these cybernetic variables, V1 representing glucose, V2 representing xylose, we can see how they change between 1 and 0 to ultimately affect change in the differential equation formulation of the model. And so, like I said before, this has been very effective in modeling or predicting various metabolic phenomena. And so perhaps you might not have an understanding of what I'll present next, but I'd be happy to talk with you more online about some of these modeling successes. And one is the prediction of multiple steady states in a reactor system. So like the other plot, this is a concentration of a metabolic product, and this is dilution rate for a reactor. And so we predicted this curve for the reactor system, and then took data points. And so what we saw is an unusual multiplicity of steady states for this reactor system. And so this is just a demonstration that the models can go beyond our current understanding of metabolism to predict things that we might not even expect to find in nature. And also, the models have been more accurate than any other modeling framework for predicting fluxes for certain growth scenarios. And this is a very complex picture of intracellular processes going on the cell. So as a seed grant team, what we're interested in doing is testing the veracity of these models. And so what we'd like to do is find new ways to apply information theory to biological data. So what we'd like to do is to take the cells, peel back the outer membrane and see what kind of data inside, what kind of intracellular processes are happening on this massive scale, and see how they converge upon these ideas of cybernetic control. So for those of you who aren't familiar with the central dogma of molecular biology, which was, this term was coined by Francis Crick, who discovered DNA back in the late 50s. it's basically the process by which we take DNA, we interpret the information that it represents, and generate some sort of biological function. So the way that this happens is that DNA is transcribed into this molecule called RNA, and that molecule in turn is functionally able to be translated into proteins. And so proteins are the functional molecules in biological systems. And so if you have something like our sugar, glucose, it'll interact with the protein and bind to that and change its, and undergo some sort of chemical reaction. And so what the protein does is make it more it can make it more energetically favorable for this chemical process to happen. And so currently, given modern experimental techniques in biology, we're able to collect massive data sets on each one of these scales. So for the RNA, we can get massive arrays of data, taking a picture of certain cellular states at a given time. And the same for protein concentration, the amount of reactants, and the rate of reactions through certain elements of the metabolic system. And so as a team, we would like to answer this question: does the sum of individual metabolic regulation elements converge upon the control goal of carbon uptake rate maximization? And so to do that, our collaboration goals are, first of all, to develop a knowledge acquisition framework for analyzing these complex sets of data. And so through that, what we would like to see is to answer that question that I posed earlier. And so some of the methods that we're currently using are to take real cells, look at their data and their gene expression changes, and then simulate those using the model to compare how the model can represent these gene expression changes. Our secondary goal is to compare our metabolic models with other metabolic models and sort of weigh them on the basis of how much relative entropy they represent in representing a real data set. And so what we would like to do is move further, and maybe you can help me when we collaborate in our breakout sessions later. But some possible alleyways that we're looking into is looking at the network context of biological data. Also, while we would prefer more deterministic methods, it's possible that there could be appropriate machine learning techniques to apply. And finally, this system might even be able to be interpreted as some sort of goal-oriented communication system. So that's all I have to say. Thank you very much.